Hello everyone, this is Mr. Sachin Kharade, Assistant Professor, Kekewa College of Agricultural Biotechnology, Nasik, going to present on the topic lysosomes and its structure and functions. So, this is the basic outline of my today's topic, that is introduction, definition, history, origin, occurrence, structure of lysosomes, polymorphisms, functions of lysosomes, and summary and conclusion. So, we will turn towards the introduction of lysosomes. So, lysosomes are spherical organelles that contain enzymes, acid hydrolases. They break up food so it is easier to digest. They are found in higher animal cells, while in yeast and plants, the same role are preferred by lytic vacuoles. So the lysosomes were discovered by the Belgium cytologist, that is Christian D. Dew, in the year 1955 and named as suicide bag or suicide sacs. So the basic origin of these lysosomes may originated from the endoplasmic reticulum and Golgi membranes from the process of budding off. So the Novikov in the year 1960 found that acid phosphatase, one of the characteristic enzyme of the lysosomes. So the occurrence of lysosomes are as follows. So the prokaryotes do not have lysosomes, but some hydrolases have been located outside the cell, as in the picture. So basically, lysosomes found in higher animals. So the picture indicate that the structure of lysosomes, so which having the lipid bilayer and the plasma membrane, and inside structure that is hydrolytic enzyme mixture. So the structure of lysosomes are as follows. So lysosomes are globular and range in diameter from zero point 0.2 to zero point 0.8 microns. Under the electron microscope, lysosomes appears dense and finely granular. So the lysosomes is essentially a sphere of lipoprotein membrane containing the enzymes, various enzymes. The membrane insulates the enzymes from the rest of the cell. If the enzymes are released, they can digest the cell, and hence lysosomes are sometimes called as suicide bags. So the lysosomal membrane are bounded by a single lipoprotein membrane. The membrane is impermeable to the substrate of enzymes contained in the lysosome. Certain substances like labrages caused in instability of the lysosomal membrane, leading to release of enzyme from the lysosomes. So other substances called as stabilizers have a stabilizing action on the membrane. Stabilizers, example vitamin A, vitamin B, vitamin K, vitamin E. So the stabilizer enzyme, example cholesterol, cortisone, and the cortisol. So the molecular weight and molecular structure plays an important part in the permeability of the lysosomal membrane. So the compound having a molecular weight higher than 200 do not diffuse through the membrane. So on the basis of molecular weight and molecular structure, this permeability of the lysosomal membrane is depends on. So this is the structure of lysosome, we can see this is the Golgi apparatus structure, and from that primary lysosomes are derived, and the after primary lysosome it will bind to the secondary lysosome and it will become fusion form of secondary lysosome. So various enzymes uh, which are present inside the lysosomes are as follows. First one is the nucleases, which include the ribonuclease and deoxyribonuclease. Break down polynucleotide into the nitrogen bases, phosphate, and pentose sugar. Next one is the phosphatases. Acid phosphatase break down phosphate esters to the monophosphate. Then the lipids, so it will degrade the lipid, break down them into the fragments. Then the protease enzyme, which are present inside the lysosomes, which break down, that is protein, break down into their constituent into the amino acid. Then the glycosides, which will break down the beta galactosidase, beta glucuronidase, then the alpha mannosidases, break down into the polysaccharide to the monosaccharides. And the sulfatase also present inside the lysosome act on the sulfate ester, breaking down into the fragments. So the polymorphism of uh, lysosomes are as follows. The lysosomes show the polymorphism in the different cell type and even within a single cell. So previously there are two types of lysosome. First one is the primary and second one is the secondary lysosome. The latter are of three types. So phagosomes, then residual bodies, and the autophagic vacuoles. So the primary lysosome is found from endoplasmic reticulum and Golgi complex. The enzymes of the primary lysosomes are synthesized by the ribosomes. It can secrete its enzyme outside the cell or store them. So phagosomes is formed by the phagocytosis and pinocytosis of foreign material. The phagosomes associate with the primary lysosome and undergoes fusion to form a secondary lysosome. So the residual bodies is the final particle containing the indigestible material it forms when digestion is incomplete. The autophagic vacuoles or autophagosomes is lysosomes containing some part of the cell in the process of digestion. So this is the different phases of lysosomes. We can see first one, this is the Golgi complex. Then from this Golgi complex, primary lysosomes will form and will fuse into the secondary lysosomes, and it will degrade the it will form a residual bodies and it will the exit from the cell. So the basic functions of lysosomes are as follows. The lysosomes is essentially a bag containing the digestive enzymes. The enzymes may released within the cell itself or outside the cell. So the lysosomes serve as several functions as follows: extracellular digestion, intracellular digestion, hormonal secretion, fertilization, then the autolysis and the chromosomal breaks. So first one is the extracellular digestion. So the digestion of extracellular matrix is a two-way process involving the both extracellular and intracellular digestion. The primary lysosomes secrete the hydrolysis into the extracellular matrix by exocytosis, resulting in partial digestion of the material of the matrix. The incomplete products of digestion are absorbed into the cell by endocytosis, and the digestion is then complete in the secondary lysosome. Extracellular digestion takes place during the bone erosion process. So the next function one is the intracellular digestion. Intracellular digestion may involve the autophagy and the heterophagy. So the autophagy, it is a intracellular digestion in which enzyme produced by the cell break down molecules and pieces of cytoplasmic material from the cell itself. So the heterophagy, it is the intake of exogenous material into the cell and the subsequent breakdown of the material by enzymes by the process of endocytosis. So heterophagy are two types, namely phagocytosis and the pinocytosis. Next one is the hormonal secretion. So there are evidence that lysosomal hydrolases are involved in a secondary activity of some cell. They functions in the modifying the product synthesized by the cell before secretion, and also in the regular of the secretion. So this is the figure of hormone secretion by the help of lysosome. Next one is the fertilization. During the fertilization process, acrosomes, that is giant lysosome of sperm head, ruptures and release of enzymes on the surface of the egg. So these enzymes digest the egg membrane and provide way for the entry of sperm nucleus into the egg. So this is the various functions in the fertilization of lysosomes we can see. Next one is the autolysis. So autolysis refers to the digestion of own cell by the lysosome. So autolysis occurs during the amphibians metamorphosis and insect metamorphosis, etc. So the autolysis by the lysosomes, which are in the figure. Then the chromosomal breaks. So lysosome contain the enzymes deoxyribonucleases, that is DNase, which attacks DNA and causes chromosomal break and rearrangement. Thus, release of hydrolases from the lysosome can result in a structural abnormalities in the chromosome that can lead to the cause of diseases like a cancer. So the summary of our today's topic is as follows. Lysosomes are small vesicles of cell organelle formed by the budding of Golgi complex and endoplasmic reticulum. Lysosome contain hydrolytic enzymes. These enzymes play an important role in the digestion. So the lysosomal enzymes are nuclease, phosphatase, lipase, protease, glycosidase, and sulfatase, and the abundantly found in the cell related with the enzymatic reactions such as liver cell, pancreatic cell, kidney cells, and the spleen cells. So the conclusion, that is lysosomes are very important cell organelle of the cell. Their prime functions to the breakup biomolecules caused because they contain the hydrolytic enzyme, but the enzymes sometimes is harmful for the cell. During starvation of organism, lysosome shows the autophagy activity and causes the cell death. Also, they cause the chromosomal abnormalities due to their DNA's enzyme. So, with this summary and conclusion, we can stop here. So, thank you for the listening.